Hello everyone, like you said, my name's Rudy. In our lab, we study the olfactory system. So I'm sure many of you are aware, but the olfactory system is what's responsible for our sense of smell. And so you may think, well, why are you studying the sense of smell? It's one of our least acute senses, and really probably a lot of people think that we would be just fine without it. While I would argue against that fact, it's not actually our perception of smell that we care about in our lab, it's actually the regenerative capabilities of the olfactory system that's of interest to us. So to highlight this, I'm going to use you all as an example. At the beginning of this semester, you all had a set of neurons that were detecting odors in the air in your nose. At this point, all of those have died. So unless you have something like a cold, like unfortunately Heath has, you probably haven't noticed a difference in your sense of smell right now. That's because even though the neurons have died, they're all replaced continuously, and that allows our sense of smell to be maintained. So looking briefly at the anatomy, you can see that I have a mouse just to show you where we're looking at, and then I have the olfactory system blown up on the right side of the screen. So in the lower part, you see what's labeled as the olfactory epithelium. This is what lines your nasal cavity and contains all the neurons that are detecting the odors in the air. And all of these neurons send projections that are called axons up to the olfactory bulb. The olfactory bulb is the area of the brain that codes all the odor information that comes from the neurons. And so for a neuron to be born, it has to send a new projection or an axon up to the olfactory bulb and establish a connection so that the sense of smell can be maintained. And this is what we care about in our lab, is how these axons are able to project and go to their specific target and establish a connection. And particularly the cells that I have highlighted in red are called ensheathing cells. And these are unique in that they have the capability, they provide an environment that allows for axons to be extended. And something that we're studying right now is they actually remove cellular debris from the neurons that have died. And this is important in reducing inflammation, and that allows those axons to be extended. And something they've shown a lot of promise in is they actually show a lot of promise in transplants for something like spinal cord injury. And this is something that's a great application for this research, is that understanding how the axons are able to extend to their target and reestablish connections can be applied to something like spinal cord injury. So that's the main goal of our research, is to be able to understand the nuts and bolts of how these axons are able to extend and what the role of the ensheathing cell is, so that one day we can provide a greater treatment for something like a spinal cord injury. Thank you.